Hope you all are doing well. Let's start solving this paper. Question number 21 says that which statement correctly describes a feature of a DNA molecule? Option A says it has a two parallel strand twisted into a double helix. We all know that DNA is having a double helix structure, but it is not having parallel strands. But it is having two anti-parallel strands, means that if one strand is running from 3 prime, there must be the other strand which must be running from 3 prime to 5 prime. Option B is saying it has complementary bases all held together by 3 hydrogen bonds. Now, this is not the case. Not all complementary bases are held together by 3 hydrogen bonds. We know that when adenine bonds with thymine, it makes 2 hydrogen bonds, and when guanine bonds with cytosine, it is forming 3 hydrogen bonds. Hence, this is also incorrect. It has phosphodiester bonds that are formed using DNA ligase. बिल्कुल, we have studied that the DNA ligase enzyme joins the Okazaki fragments to form a complete strand. For example, these are the two lagging strands which were formed by the DNA polymerase, and then these two lagging strands या फिर जो है Okazaki fragments are joined together by the DNA ligase enzyme. So hence the answer over here is option C. We can also check out option D, that why option D is not our correct answer. It has a backbone of phosphate molecules and nucleotides. No, this is not the correct option. Why? Because there is a sugar phosphate backbone. In DNA molecule, the backbone is known as the sugar phosphate backbone, which is also having phosphate molecules as well as the sugar molecule. Number 22 over here says that which diagram correctly shows the replication of a DNA. If I talk about the replication of the DNA, we know that the essential या फिर vital enzyme which is needed for the replication is the DNA polymerase. And this is the enzyme which makes the new DNA strand in the direction of the 5 prime to 3 prime. If I explain this further, we know that the DNA structure is a double helix structure. And if I talk about the strands over here, I know that let's assume that this strand, the blue one, is from 5 prime to 3 prime over here. And the red strand over here is from 3 prime to 5 prime over here. Now let me draw this over here so that we can show the semi-conservative replication of the DNA. Right? Now these are the two strands which I have shown over here. One is the red strand and one is the blue strand from the DNA. Now what is going to happen that this is the old strand. Now what is going to happen that now what will happen that the DNA polymerase is going to synthesize the Now what is going to happen over here is that DNA polymerase is going to synthesize the strand from here in the direction of 5 prime to 3 prime, which means that the new strand which will be formed over here will be from the direction of 5 prime to 3 prime. And then similarly over here in the red part, the DNA polymerase will start to replicate the DNA from this strand. And it will go like this. From 5 prime to 3 prime, it is going to make a new strand. Right? So अब जो है, if I discuss about it, so we know if this is 3 prime and this is 5 prime, and the new strand that is going to be formed over here should be from 5 prime to 3 prime, and DNA polymerase replicate it from 5 prime to 3 prime. So S2 is showing us the correct direction of the replication. And if I talk about S1, we know that this is from 3 prime to 5 prime, and we over here know that what will happen, that the DNA polymerase will be over here and it will start to replicate this strand like this from 5 prime to 3 prime. So over here the answer is option C. So question number 24 over here says that four students sketch the diagrams of the apoplast pathway and the symplast pathway. Which sketch is the most accurate diagram of the two pathways? To solve this question, we must have the knowledge that what is the apoplast pathway and what is the symplast pathway. We know that symplast pathway is that pathway in which there is a passage of water from cell to cell. In this pathway, the cytoplasm and the cell surface membrane is also involved. और apoplast pathway हमारे पास it is that passage of water which happens via the cell wall and the intracellular air spaces. So if we see over here, we can say that this is the apoplast pathway where there is the passage of water via the cell wall only. At this cell, which is the endodermis cell, there is a Casparian strip. This is a waxy strip. What does this do? That it stops the passage of water via the apoplast pathway. So the water has to change its direction and enter the endodermis cell via the symplast pathway. From here, now the water is traveling via the symplast pathway. And this is the symplast pathway in which the water is traveling via the cell to cell through the cell surface membranes and the cytoplasm as well. So hence the answer to this question is option D. Question number 25 over here says that the photomicrograph shows some xerophytic adaptations in the leaf tissue. We know that xerophytes are the plants which are adapted to live in the minimal supply of water. Which row shows the correct functions for the structure 1, 2 and 3? First of all, let us start labeling the structure 1, 2 and 3. Structure 1 over here is basically representing the trichomes. Structure 2 is representing the rolled leaves. And structure 3 over here is basically representing the thick waxy tissue. Now, if we begin with the function for the trichomes and the rolled leaves, we know that trichomes and the rolled leaves are adapted in a way that they trap the saturated air and water vapors within them. Because of this, what happens that it creates a humid environment around the stomata of the leaves, जिसकी वजह से the water potential gradient between the air spaces, the intercellular air spaces and the atmospheric air decreases. Due to which the rate of transpiration also decreases and hence the loss of water from the leaves is also less. So over here what can we say that traps the moist air? Traps a layer of moist air. तो हमारे पास क्या ऑप्शन होगा that 1 and 2 both traps the layer of the moist air. अच्छा thick waxy cuticle की अगर हम लोग फंक्शन की बात करें so thick waxy cuticle is having the function that it prevents the excessive water loss. Why? Because it is waterproof, right? And as it is waterproof, it prevents the excessive water loss. So reduce the evaporation from the epidermis of the leaf. We know that all the three structures are playing the role in reducing the evaporation from the epidermis of the leaf. Reduces the water vapor concentration gradient between the inside and the outside of the leaf. We have already discussed over here that structure 1, the trichomes, and structure 2, the rolled leaves, are basically reducing the water vapor concentration gradient between the inside and the outside of the leaf, due to which the rate of transpiration decreases and more water can be saved by the plant. Hence, the answer over here is our option B. Question number 26 over here says that which processes are required for the transfer of assimilates from the source cell into the phloem sieve tube. So in order to understand this question, first of all, what we are going to do is that we are going to understand the process in which there is the loading of sucrose into the phloem sieve tube elements. So first of all, what happens that the companion cells are using the ATP to pump the hydrogen ions from the companion cells into the cell wall, right? So hydrogen ions concentration increases inside the cell wall. The process through which the companion cells are transporting the hydrogen ions into the cell wall is by the active transport. Right, and when the hydrogen ion concentration inside the cell wall increases, what happens is that the hydrogen ions then diffuses back into the cell wall, into the companion cell by the co-transport protein. And when the hydrogen ions are diffusing back down the concentration gradient with the help of a co-transport protein, so we know that whenever the diffusion uses a co-transport protein, the facilitated diffusion is said to be the process over there. And we know that when the hydrogen ions diffuses down the concentration gradient, it basically releases the energy to pump the sucrose from the companion cell, from the source cell into the companion cell via the same co-transport protein down against the concentration gradient. And when it is happening against the concentration gradient, we know that it is the secondary active transport. As the sucrose concentration inside the companion cell increases, then what will happen that the sucrose will diffuse from the companion cell to the sieve tube element via the plasmodesmata. So what is happening over here? Active transport is the process that is happening over here. Facilitated diffusion is the process that is happening over here, and simple diffusion is the process that is happening over here. So option A will be our correct answer. Question number 27 over here says that when the sucrose is loaded into the phloem, it has to travel from mesophyll cell to a companion cell and then into the phloem. Which row shows the relative concentrations of sucrose in each type of the cell in order for this process to take place? We all know that sucrose which is made in the mesophyll cell is transferred to the companion cell via the process of the active transport from a low concentration to a high concentration. And then when the sucrose reaches the companion cell, it then diffuses into the phloem sieve tube element via the plasmodesmata. So, the transfer of sucrose from the companion cell to the phloem is done by the process of the diffusion. Diffusion is from the higher concentration to the lower concentration. So, from here we can conclude that the companion cell should have a higher concentration of the sucrose than the mesophyll cell. Why? Because the active transport is taking place. So this is the case in option A and in option B. We will exclude option C and D because it is not fulfilling the active transport scenario. Then we know that from the companion cell to the phloem sieve tube element, we know that diffusion process takes place and diffusion is from the higher concentration to the lower concentration. This is the higher concentration of the companion cell and this is the lower concentration in the sieve tube element. So hence the answer over here is option B. Question number 28 over here is saying that which feature of transport in plants is correct for the xylem and phloem. Now let's one by one look at each option and try to eliminate the wrong option. It is passive. Xylem में the transport is passive, but in phloem we know that the loading process and the unloading process is active. They both are the active process. Let's look at option C. It occurs from source to sink. We know that the transport in the phloem can occur from source to sink, but the transport in the xylem occurs from the roots to the leaves only. Option D over here is saying that it occurs only in one direction. We know that in the xylem, the transport of the water occurs in only one direction, but whereas the transport in the phloem is said to be bidirectional, which means that it occurs in both the directions. So over here our correct answer is option B. It occurs by the mass flow. We know that the mass flow is common in both the xylem and the phloem. Question number 29 over here says that the diagram shows an interior view of a vertical section through the human heart. Which row shows the correct labels for the diagram? So first of all, let us quickly label the whole diagram of the heart over here. We know that this part over here is the aorta. This over here, which is the structure 4, it is basically indicating the vena cava. This is over here the right atrium, which receives the blood, deoxygenated blood from the vena cava. Then this is the right ventricle. And over here it is connected by the walls, which walls are known to be the tricuspid walls. Then let us move to the other side of the heart. This structure 1 is basically, it is below the aorta and it is representing the pulmonary artery, which pumps the deoxygenated blood to the lungs. This structure over here is representing the left atrium, and this structure over here is representing the left ventricle. We know that in between the left atrium and the left ventricle there is a valve, and that valve is known as the mitral valve या फिर bicuspid valve. We know that the structure 2 over here is basically representing the pulmonary valve, and this valve over here is basically known as the aortic valve, which is basically between the left ventricle and the aorta. So now let's just see that what is the structure 1, 2, 3 and 4 representing. Structure 1 over here is representing the pulmonary artery. Structure 2 over here is representing the pulmonary valve, which is also known as the semilunar valve. This is correct. Semilunar valves are the valves which are found between the ventricle and the arteries. Then the structure 3 is representing the tricuspid valve. Tricuspid valves are also known as atrioventricular valves, which are basically found between the atrium and the ventricle. The structure number 4 is representing the vena cava, which receives the oxygenated blood from the rest of the body. So over here, our answer is option C. Question number 30 over here says that the inner layer of veins and the capillary contain. We know that the structure of capillary is extremely simple. It contains a layer of the endothelial cells which are resting on a basement membrane. Then if we talk about the structure of the vein, we know that in vein there are three layers of the tissues. One is the tunica externa, the second is the tunica media, and the third layer over here is known as the tunica intima. This layer of the tunica intima is made up of the endothelial cell. The tunica intima only contains the endothelial cell. So what does the inner layer of the veins and the capillary contain? Capillary only contain the endothelial cell, and the inner layer of the veins is also containing the endothelial cell, which is known as the tunica intima. This layer is also known as the tunica intima. So over here our answer should be option C, endothelium. Also, there is one more common MCQ which is asked in the paper, that what remains constant in all of the three blood vessels. So, the thickness of endothelium remains constant in all of the blood vessels. The thickness of the endothelium does not changes in any of the blood vessel, whether it is the capillary, whether it is the vein, or whether it is the artery. Question number 31 over here is saying to us that the graph shows the changes in the blood pressure and the water potential across the blood capillaries from the arterial end of the capillary to the venous end of the capillary. So over here they are showing us the graph of the arterial end, which is starting from the arterial end which is ending at the venous end. We have been shown that arterial end से लेके venous end तक the blood pressure is decreasing. Now they have asked that what could result in the accumulation of excess tissue fluid. We know that the condition in which there is the excess tissue fluid is known as the oedema. We have studied the conditions, या फिर we have studied the factors which causes the oedema. What are the conditions that are causing it? It is the blockage of the lymph. We know that if the lymph vessels will be blocked, then the excess tissue fluid will not be drained. The second factor is that the protein decreases in the blood. We know that as the protein concentration in the blood decreases, what is going to happen that the water potential in the blood is going to increase. And as the water potential in the blood is going to increase, so there will be less tissue fluid which will return to the capillary as the water potential gradient is not maintained. Less fluid into the blood capillary. Then the third factor over here is the high blood pressure, which is known as the hypertension. It is also one of the causes of oedema, which is the accumulation of the excess tissue fluid. We know that when the blood pressure becomes more high, then it is at the arterial end. What will happen that more fluid will leak out of the capillary at the arterial end, more fluid than usual. So what will happen that more fluid will leak out at arterial end. So now let us look at the options and let's see that what is our correct answer. An increase in the concentration of large proteins in the blood plasma. We know that if there would be an increased concentration of proteins in the blood plasma, the water potential would decrease. And if the water potential will decrease, then more so what will happen that more tissue fluid is going to flow back into the capillary. So this will not result in the accumulation of the tissue fluid. An increase in the blood pressure. Yes, we have discussed this reason over here. Statement 3, which are the factors of the oedema, में हमने ये चीज discuss किया that high blood pressure is the cause of the accumulation of excess tissue fluid. So hence our answer over here is option B. If I look at the other options as well, so we know that decrease in the blood pressure तो हमारा आंसर नहीं हो सकता. Why? Because increase in the blood pressure is causing the accumulation. So decrease in the blood pressure will not cause the accumulation of excess tissue fluid. If we look at option C, it says that a decrease in the concentration of small proteins in the tissue fluid. If the small proteins in the tissue fluid will decrease, तो हमें पता है that the water potential over there will increase. And if the water potential of the tissue fluid will increase, so we know that it will flow back to the capillary. And hence there will be no accumulation of excess tissue fluid. So this is also an incorrect statement for this question. So hence our answer over here is option B. Question number 32 over here says that which statement is correct about the red blood cell that has just entered the capillary in the respiring muscle. We know that red blood cells are having the function to transport the oxygen. Respiring muscles have a higher partial pressure for the carbon dioxide, and because of this high partial pressure of carbon dioxide in the respiring muscles, the oxygen is dissociated from the red blood cell. As this oxygen gets dissociated from the red blood cell, what will happen that the carbon dioxide is going to bind with the amine group at the polypeptide chain of the hemoglobin in the red blood cell, and it is going to form carb amino hemoglobin. So in this, they are asking that what is the correct statement about the red blood cell which has entered a respiring muscle. So we know that there is a net increase in the amount of carb amino hemoglobin as the carbon dioxide is going to combine with the amine group of the polypeptide chain of the hemoglobin. Hence our answer over here is option D. Here we are with question number 33. What this question number 33 over here says, the graph shows some blood pressure changes that occurs in a human during one cardiac cycle. This is the graph. It is showing the one cardiac cycle. It says that when does the systole starts and ends in the right atrium and in the right ventricle. In order to solve this question, we must first interpret the graph given over here. We know that this is representing the atrial systole, this is representing the ventricular systole, and this over here is diastole. We know that in diastole both the atrias and the ventricles are relaxing. This is the ventricular systole over here and this is the atrial systole over here. We know that in the atrial systole the atrias are contracting and in the ventricular systole the ventricles are contracting. Now let's see that in the right atrium, where does the systole starts? We know that this is the atrial systole over here and it is starting at the point T and it is ending at the point V. In the right ventricle, we can see from the graph that the systole is starting at the point where the atrial systole is ending basically, which is the point D over here. And here this is where the systole ends. We can see from the graph that the systole is ending at the point X. Hence the answer for this MCQ over here is option A. Now let's have a look at question number 34. It says that this statement about all bronchioles are correct. In order to do this question, we must have a look at the cross-section structure of the bronchioles. We know that the cross-sectional structure of the bronchioles, if we have a look at it, we know that this arrow is indicating the lumen over here. This innermost structure is the ciliated epithelium without the goblet cell. Then this layer over here, which I am highlighting, is basically the connective tissues which are without the mucus gland. So from here, we can conclude that the mucus gland and the goblet cells are not a part of the bronchioles. Then over here, this outermost structure, here outermost layer is basically the smooth muscle, right? So they have the goblet cells. No, this statement is incorrect that they do not have the goblet cells within them, as we know that ciliated epithelium layer is without the goblet cells. They have the epithelial cells. Yes, this is correct because the innermost layer we have studied is having the epithelial cells. To be more specific, they are having the ciliated epithelial cells. They have the muscle tissue. Yes, they are having the muscle tissue, which is the outermost layer. So hence our answer over here would be option D, 2 and 3 only would be our correct answer. Question number 35 over here says that what is shown in the electron micrograph. We know that if we talk about the electron micrograph, there are two types of electron micrograph. One is the scanning electron micrograph and the other one is the transmission electron micrograph. The basic difference between these two micrographs is that in the scanning electron micrograph, we can just see the surface of the cell, whereas in the transmission electron micrograph, we can see the interior of the cell as well. So they are over here asking that what is shown in this electron micrograph. We know that this cannot be the transmission electron micrograph because we can clearly see that the surface of some of the cells are being shown in the above diagram. Now it can be a scanning electron micrograph, right? This is the scanning electron micrograph of the lung squamous epithelium and the red blood cell. These over here are the red blood cells and this whole structure over here which I am highlighting with yellow is basically the lung squamous epithelium. So hence the answer over here would be option B. It is showing the scanning electron micrograph of a lung squamous epithelium and the red blood cell. Question number 36 over here is saying that this statement correctly describes the infectious diseases. In order to interpret this question, we must have the knowledge of the infectious diseases. We know that infectious diseases are the diseases that are caused by the pathogen and they are transmittable. or we can say that they can be transmitted as well. What are the pathogens that may cause the infectious diseases? They are the bacteria, they can be the virus, they can be the fungi. Now let's look at the statements one by one and let's see that which is the correct statement. They are the diseases caused by the environmental factor and are not passed from one person to another. No, these are the diseases that are caused by the pathogens which we have studied over here, and they are transmittable. So these both statements are incorrect. They are the diseases caused by the fault in the DNA. No, this is incorrect. They are the diseases caused by the pathogen. They are the diseases caused by the lack of nutrition in the diet. No, this is as well the wrong statement. Option D is saying they are the diseases that are caused by a pathogen such as bacterium, fungi, protoplast, and the virus. So yes, this statement over here, which is the option D, satisfying the infectious diseases. So hence our answer over here is option D. Let's see question number 37. It says that organism X produces molecule Y which blocks the activity of the 70S ribosomes. An experiment was carried out in order to investigate the effect of Y on the bacterial cells. On agar plate with bacteria growing all over its surface had four walls cut into the agar. Different substances were added to each well and the agar plate with bacteria was incubated for a week. The diagram shows the results after a week. They are then asking in the question that what conclusions can be made from these results. We know that this arrow over here is in the sterile distilled water condition. This is the intact cells of the organism X. This is the commonly used antibiotic. This is the concentrated solution made from the cytoplasm of a lot of organisms of X cell. Now they are saying that what conclusions can be made from these results. So we will have to use this figure in order to tell that what is the conclusion which is being made from the results. Molecule Y function as an antibiotic. We know that yes, molecule Y can function as an antibiotic. Why? Because you can over here see that there is no bacterial growth over here as well and over here as well. So molecule Y can function as an antibiotic. Molecule Y cannot be released from the organisms of Molecule Y cannot be released from the cells of organism X. Yes, this is being shown over here that this is the intact cell and intact cells are those cells that are unaffected by a disease. So we know that molecule Y cannot be released from the cells of organism X. This is also a correct statement over here. Then molecule Y would also affect the synthesis of protein from the nuclear DNA in the human cell. No, this is not the case. Why? Because it is not being shown in the experiment above. We cannot conclude this statement from the experiment above. So hence the answer over here is option B. Question number 38 is saying that a patient has had repeated infections from the same pathogen over a few months. The patient has been treated with the same antibiotic on each occasion. Which treatment could prevent an increase in the antibiotic resistance of the pathogen? If we talk about the antibiotic resistance, we know that it is a condition in which a particular antibiotic becomes ineffective against a specific pathogen or a species of pathogen. Now they are saying that we need to prevent an increase in the antibiotic resistance. So they are asking that how we can prevent the increase in that antibiotic resistance. They are saying in option A, a narrow spectrum antibiotic to treat a non-cellular pathogen. If we are talking about a non-cellular pathogen, we know that the non-cellular pathogens can be the viruses, they can be the prions, and we know over here that the antibiotic only are effective against the bacterial infections. And narrow spectrum antibiotic are those antibiotics which acts at a specific groups of bacterial cells. And antibiotics are only effective against the bacteria. So this cannot be the option A is not our correct statement. A repeated prescription for the same antibiotic at a higher dose. We know that if we are repeating the same antibiotic for the same infection over and over again, it will increase the chances for the antibiotic resistance. So this is also not our correct option. A specific antibiotic after testing for the pathogen. Yes, if there is a specific antibiotic which is only affecting a particular pathogen which is causing the repeated infection, then we know that this will decrease the chances for the antibiotic resistance. So hence the answer over here is option C. If we look at option D, a wide spectrum of antibiotic to treat the different strains of the pathogen. We know that when we will use the wide spectrum of antibiotic to treat the different strains of the pathogen, what will happen that even those pathogens that are not involved in the repeated infection will get introduced to the wide spectrum of the antibiotic. And what this is going to do is that this is going to increase the chances for the antibiotic resistance in the other bacteria as well. So hence the answer over here is option C. Question number 39 over here is saying that a patient with kidney failure can be given a kidney transplant. In a kidney transplant, a healthy kidney is taken from another person and put into the patient's body. Sometimes the donated kidney cells are killed by the patient's body. In this, the primary immune response, T lymphocytes bind to the antigen on the donated kidney cells and are activated. Which statement correctly explains how the donor kidney cells are killed? So this basically is the question about the transplant rejection. And over here they are also talking about the primary immune response. We know that as the T lymphocytes bind to the antigens on the donated kidney cells and are activated, what is the next step over here that the activated T lymphocytes then secrete the chemical which is known as the cytokine. What does these chemicals over here do is that they stimulate the T lymphocytes in order to do the clonal expansion. And what happens in this clonal expansion is that the T lymphocyte cells are dividing by the process of the mitosis and they are increasing in their number. Then some of the T lymphocytes differentiate into the T killer cells and some of the T lymphocytes are differentiated into the T helper cells. Over here as the over here as there is the rejection happening and the cells are being killed. So hence over here the T killer cells are being produced. Then what will happen after the differentiation of the T lymphocyte cells into the T killer cells? After that what will happen that the T killer cells are going to bind to the donor kidney cells. And as the T killer cells are going to bind with the donor kidney cells, the T killer cells are going to release the toxins that destroy the donor's kidney cell, and within that toxin there must be the granzymes and the perforin. What do the perforins do? Perforins basically make the perforate the cell membrane or it basically makes the holes in the cell membrane. And what are the granzymes and what do the granzymes do? Granzymes are the enzymes that enter the cell and destroy them. So what is the correct sequence over here which is telling us the path of the primary immune response which is be followed by the T lymphocytes? It will be that first the activated T lymphocytes are going to secrete the cytokines which are basically the chemicals. Then what will happen that these chemicals will stimulate the T lymphocytes to multiply by the mitosis. Clonal expansion will take place and then those T lymphocytes are going to differentiate into T killer cells or the T helper cells. Over here as there are the kidney cells which are killed, the kidney cells are killed, so hence the T killer cells will be produced. Then what is going to happen that the T killer cell are going to bind to the antigen of the donor's kidney cells, and then when the T killer cell will bind to the antigen, what will happen that the T killer cells are going to release the toxins, and that toxins can be the enzymes as well as the perforin. Okay, so over here our answer is option B. Now let's discuss the last MCQ of this paper. MCQ number 40. The diagram shows the simplified structure of an antibody. We know that the structure of the antibody consists of two heavy polypeptide chains and it consists of two light polypeptide chains. If we discuss about the structure U and structure Z over here, we know that this is representing the variable region of the antibody. What does this region do? That this region is specific to a particular type of antigen and only a particular or a specific type of antigen can bind to this site. This can also be said to be the binding site for the antigen. If I discuss about the structure V over here, this structure V over here is basically the hinge region. What does this region do? That this region provides the flexibility to the antibody molecule. Why does it provide here? What is the significance of it providing the flexibility to the antibody molecule? Is that so that the two binding sites over here, which is X and Y, they can make contact with the antigens that are separated by a slightly different distance. Also, if I discuss over here about the disulfide bonds, these are the disulfide bonds which are found between the two heavy polypeptide chains of the antibody. What is the function of these disulfide bonds over here? The function of these disulfide bonds over here is that they hold the antibody together. They are holding the structure of the antibody together. They are asking that which statement is correct. We know that structure V, which is the hinge region over here, allows the antibody to be flexible so that the two binding sites over here, which is U and Z, can make a contact with the antigens that are slightly different distances apart. So hence our answer over here would be option B.